Ladies and gentlemen, please rise. The Honorable Senate is entering. The university flag will be now brought in. Ladies and gentlemen, please be seated. I now invite Professor Łukasz Sułkowski, President of WSB University, to address the Assembly.
Ladies and gentlemen, it's a great honor to declare the ceremony of commemorating the conferment of the title of Doctorate Honoris Causa of WSB University open. With the deepest respect, I extend my warmest welcome to all of you and express my sincere gratitude to you for joining on this special occasion today. Ladies and gentlemen, the title of Honoris Causa is the highest distinction that a contemporary university may grant upon its celebrities and the most important members. It's a great honor and privilege for us from WSB University that the title of Doctorate Honoris Causa is being conferred upon Professor David E. Fisher, a world-renowned scientist in the field of medicine, molecular biology, oncology, and outstanding ambassador of science worldwide, and a man who embodies the highest academic values and traditions. With profound respect, I extend my heartfelt welcome to the distinguished recipient of our honorary doctorate, the internationally renowned researcher, clinician, and academician, head of Massachusetts General Hospital Department of Dermatology at Harvard Medical School in Boston, director on MGH Cantonese Biology Research Center and director of Melanoma Center at MGH, professor of dermatology and pediatrics at Harvard Medical School, Professor David Erich Fisher, PhD, a scientist, a great passion and dedication, a man of remarkable intellect. We warmly welcome Professor David Erich Fisher here at WSB University. It is also great pleasure to extend a cordial welcome to Dr. Claire Fang Fisher, Professor Fisher's wife, a radiologist, oncologist at Beth Israel Deaconess, She receives her medical degree from Harvard Medical School and has been practicing for over 20 years. Welcome very warmly here in Poland at WSB University. I also wish to extend a very warm welcome to the reviewers in the procedure for the conferment of the highest academic honor of WSB University, Professor Lidia Rudnicka, a distinguished dermatologist and venerologist, honorary president of the Polish Dermatological Society, head of Department of Dermatology at Medical University of Warsaw. Welcome, Professor. And Professor Aaron Ciechanover, biochemist and Nobel laureate in chemistry, distinguished researcher, professor in the Faculty of Medicine at the Technion, Israel Institute of Technology. He is internationally renowned for the groundbreaking discovery in a protein degradation, a fundamental process in biology. Welcome. The role of the supervisor in the procedure for the conferment of the highest academic honor at WSB University is fulfilled by Professor Dr. Tomasz Gąsior, associate professor at WSB University. This is the function of rector proxy at the development of the Collegium Medicum at WSB University, the recipient of the Ministry of Science and Higher Education Scholarship for Outstanding Young Scientist, recipient of the Prize of the Politics for Science in Health Sciences, an eminent cardiologist, researcher with publications in prestigious scientific journals, collaborating with Zurich University, University of Cambridge, and Harvard University. Welcome, Professor. On behalf of the parliamentary representatives of the Republic of Poland, I am honored to welcome Mr. Mateusz Bochenek, member of the Polish Parliament. Welcome. I also extend a warm welcome to the representatives of the public health institutions and social institutions, Dr. Piotr Grzesik, member of the Presidium of Silesian Medical Chamber in Katowice. Welcome, Professor. I warmly welcome the representatives of the state institutions and public administration, Mr. Piotr Kalbron, District Labor Inspector in Katowice. Welcome. Deputy Commissioner Robert Szewczyk, Deputy Head of the Małopolska Custom of the Revenue Office. Welcome. On the behalf of the rectoral and dean authorities and the WSB University Senate, I extend a warm welcome to Professor Katarzyna Szczepańska-Woszczyna, Vice Rector for Science and Education, Member of Council of Scientific Excellence, and expert of Polish Accreditation Committee. I would like to also welcome our authorities of Collegium Medicum, Dr. Adam Kabierz, Dean of the Collegium Medicum at the WSB University. Welcome, Professor. I would like to welcome Dr. Monika Lis, Dean of the WSB University Branch Campus in Krakow. Dr. Dariusz Trojszczak, Dean of WSB University in Żywiec. And Dr. Justyna Misiak, Deputy Dean of the Collegium Medicum at WSB University. Welcome. I also extend a warm welcome to the representatives of the students of the Collegium Medicum of WSB University. Welcome. Ladies and gentlemen, once again, I warmly welcome all of you. I extend my heartfelt welcome to the members of the WSB University Senate members, vice rectors, deans, deputy deans, faculty members, professors, assistants, doctoral candidates. I'm also very pleased to welcome the representatives of the partner universities and all distinguished guests and friends of our university. Welcome. In accordance with academic tradition, I hereby solemnly declare the ceremony commemorating the conferment of the title of honoris causa of WSB University upon Professor David Fisher formally open. Thank you.
Thank you, President. Ladies and gentlemen, now I kindly invite Professor Lidia Rudnicka, the reviewer in the procedure for awarding the title of Doctor Honoris Causa, to present her review.
Hello, dear Rector, distinguished guests. I would like to thank for the invitation to prepare this review. Also, a big thank you to the university for translating the review into English. I'm sorry, I thought that the celebration today will be in Polish, so I prepared my original review in Polish. I will now read significant elements of the English version of this review. Professor David Fisher is an outstanding researcher and oncologist affiliated with the Harvard Medical School. His scientific achievements had a significant impact on the development of medical knowledge, especially in the area of melanoma. It was concentrated on the pathogenesis of melanoma and on finding new targets for therapy. Professor Fisher published the results of his studies in highly impacted and highly prestigious scientific journals such as Nature, Science, Cell, and they have also been intensely cited, showing the very strong impact that his work had on medical knowledge. Some of his most important work includes the research on MITF, which plays a key role on how melanocytes turn into melanoma cells. This discovery that was published in 2005 facilitated the understanding of how MITF may control the genes which are related to the growth of melanocytes and of development of cancer, and specifically in this case of melanoma. Another big article of significant importance was published in Cancer Cell in 2013, and it was concentrating on the BRAF gene and how BRAF may affect cancer cell metabolism through selected molecules. Professor Fisher also studies the effect of UV light on the development of melanoma. And while it is known for a while that sunburns and excessive exposition to UV may cause melanoma, this research helped to explain what are the molecular mechanisms behind this phenomenon and what mechanisms are responsible for the conversion of normal melanocytes into melanoma cells. Professor Fisher's team explored how UV radiation may cause mutations that lead to cancer and how these mutations may affect how they may be a target for the future development of therapies. A significant part of the studies was developed also to melanoma vaccines and to shaping an even more personalized way of treating melanoma. The global importance of studying and publishing findings on the link between UV and melanoma cannot be really overrated. These studies illustrate the molecular pathways by which UV exposure damages DNA in melanocytes, leading to the mutations that drive the development of melanoma. This insight is really important for the public health initiatives as it equips the physicians with arguments for their campaign and awareness programs. And I must add, in addition to the written evaluation, that such studies have an essential effect or important effect on putting in 2025, just a few months ago, sunscreens into the essential medicines list of the WHO. And also, would like to emphasize that the WHO has just recently established a resolution or adopted a resolution on the importance of skin health and skin diseases as a priority in human health. This is of significant importance for the future development of dermatology. But further to the evaluation, in 2014, Dr. Fisher and his co-workers published a landmark review in Science, and it was entitled "The Melanoma Revolution from UV Carcinogenesis to the New Era of Therapeutics." This article provided a comprehensive overview of the evolution of melanoma research from the early studies on UV-induced carcinogenesis to the emergence of new modern therapies. And this article has multiple citations, and it has become a very important overview in the field of cutaneous oncology. Additional also interesting research was published in 2014. It explored the role of beta-endorphin in mediating UV-related behaviors, offering the insight into the psychological and physiological factors that influence the individual's use of artificial UV sources, but also excessive exposure to natural UV light, despite all the known risks. So we thought it's psychological. Now it is explained how physiology may contribute to the need of some people to become exposed to UV light over and over again, despite all possible risks. More recently, Dr. Fisher has co-authored an article which I found especially interesting on the immunotherapy and target therapies of melanoma and other skin cancers. And I liked especially the article which was on a topical treatment of giant congenital nevi. Giant congenital nevi are associated with a high risk of development of melanoma. And the study shows that there is a topical agent, a topical medication that may not only cause or induce some involution within the congenital nevi, but it may also decrease the risk of melanoma in these patients. So in conclusion, David Fisher is a researcher with substantial record which combines an interdisciplinary linking approach of molecular biology and clinical practice. His publications have a significant impact on the development of medical knowledge and are characterized by highest quality and innovation, and from my perspective, most importantly, by their potential of translating research findings into clinical practice. Professor Fisher fulfills the requirements of your university, of WSB, regulation for awarding the Doctor Honoris Causa award, and there's no doubt that he fulfills the requirements of these regulations by being a scientist of exceptional merit to science. And I may also add that before I accepted to make this review, I had the opportunity to contact and communicate with friends who are dermatologists at Harvard Medical University. And I must say that the feedback was highly positive, indicating that Dr. Fisher is an extremely positive person in regards to excellence in science and the ambitious goals. So it was a pleasure and an honor to prepare this review. Thank you very much.
Thank you, Professor, for your opinion and recommendation. Thank you. And now I invite Professor Tomasz Gąsior, the supervisor in the procedure for awarding the title of Doctor honoris causa, to deliver his laudation.
Your Magnificence, distinguished professors and representatives of the academic community, esteemed guests, ladies and gentlemen, it is a profound honor to stand before you on this remarkable day, a day on which the WSB University celebrates knowledge, discovery, and the highest values of academia. Today, by conferring the title of Doctor honoris causa upon Professor David Fisher, we honor not only an exceptional scientist, but a visionary whose work has transformed medicine, whose leadership has shaped institutions, and whose humanity has inspired generations of scholars and physicians around the world. Professor Fisher's academic journey has been exceptional from the very beginning. He studied chemistry and biology at the Swarthmore College, earned his PhD at the Rockefeller University under the mentorship of Nobel laureates Gunther Blobel and Henry Kunkel, and later completed his medical degree at Cornell. His clinical training continued at MGH, Massachusetts General Hospital, followed by fellowships at Dana-Farber Cancer Institute and Boston Children's Hospital, and postdoctoral research with Nobel laureate Philip Sharp at Massachusetts Institute of Technology, MIT, creating a rare and powerful union of fundamental science and medical practice. It's really hard to think of a better academic environment than getting degrees at Ivy League and pursuing the academic and medical career at the Boston area. Today, Dr. Fisher serves as a Lancer Professor at Edward Wigglesworth Distinguished Professor of Dermatology at Harvard Medical School and a Chair of Department of Dermatology at MGH, one of the most respected academic medical centers in the world. He also directs cutaneous biology research center and melanoma program at the MGH Cancer Center and the US Department of Defense Melanoma Academy, shaping the future of career research on a national and global scale. At the heart of Professor Fisher's scientific legacy is his groundbreaking research or work on MITF, the transcription factor, the master regulator of pigment cells, and a key oncogene in melanoma. His discoveries revealed how this molecule governs both normal pigmentation and cancer progression, and this insight continues to guide precision oncology across the world today. He also changed the way we understand skin cancer risk. His lab showed that pheomelanin, the pigment responsible for red hair, can generate oxidative damage even without ultraviolet light. This was a profound shift in thinking. Cancer risk is not defined only by the environment, but also by intrinsic biology. Building on that knowledge, his team introduced a visionary strategy for prevention. Pharmacologic tanning without UV exposure, offering protection for individuals at the highest risks. In the field of cancer immunology, as mentioned before, his laboratory has revealed how melanoma can evade immune system and how that invasion can be overcome. He has shown that immune checkpoint inhibitors can be made even more effective when combined with local therapies, such as fractional laser treatment, work that is already shaping clinical trials and improving outcomes for patients around the world. Professor Fisher's curiosity has also led him into a fascinating connection between the skin and the brain. His team discovered that ultraviolet light stimulates endorphin production in the skin, activating opioid pathways in the brain, essentially explaining why some people seek sunlight almost like an addiction. This changed the way we understand tanning, not merely as a cosmetic habit, but also as a neurobiological drive, an intrinsic need. His discoveries have been published in the world's most prestigious scientific journals, including Nature, Science, Cell, and the New England Journal of Medicine. Beyond his scientific achievements, Professor Fisher is an institution builder and a mentor of extraordinary impact. Since 2008, he has led the Department of Dermatology at MGH, establishing a culture where clinical care, research, and education are deeply interconnected, deeply united. Under his leadership, the department has become a global model for translational medicine, and many of his former trainees now lead laboratories, medical centers, and biotech initiatives across the globe. At Harvard College, he's also been a highly respected educator. His acclaimed undergraduate course, Skin, Your Largest, Hottest, and Coolest Organ, has inspired many young people to pursue their careers in medicine and in dermatology. His achievements have been recognized at the highest level. He's been elected to the United States National Academy of Medicine, and most recently named as a fellow of AACR Academy, one of the most prestigious honors in cancer research. And yet, Professor Fisher's life cannot be fully understood without music. Trained as a cellist at the Curtis Institute of Music, he made his Carnegie Recital Hall debut to highest professional recognition and has performed at Carnegie Hall, the Kennedy Center, and the Philadelphia Academy of Music under the direction of legendary conductors including Eugene Ormandy, Zubin Mehta, Riccardo Muti, and Claudio Abbado. The discipline required in classical music, the concentration of chamber performance, and the constant pursuit of harmony are the same qualities that define science: precision, collaboration, and the tireless search for truth. Ladies and gentlemen, I'm honoring Professor Fisher. In honoring Professor Fisher, WSB University honors a person who truly embodies the unity of science, medicine, education, leadership, and art. His discoveries have redefined how we prevent, diagnose, and treat disease. His leadership has strengthened institutional and international collaborations. His teaching has awakened curiosity and courage in young minds. And his music reminds us that knowledge, when guided by imagination and purpose, becomes art in the service of humanity. Therefore, with great gratitude and admiration, WSB University confers upon Professor David Fisher the title of Doctor Honoris Causa. With profound respect, congratulations, Dr. Fisher.
Thank you, Professor. Ladies and gentlemen, please rise. We are now commencing the main part of the ceremony, commemorating the conferment of the academic honor of Doctor Honoris Causa upon Professor David Fisher. I invite Professor Katarzyna Szczepańska-Woszczyna, Vice Rector for Science and Education, to read the text of the diploma of the distinguished Doctor Honoris Causa. The university flag come out.
On behalf of the Director of WSB University, Professor Zdzisława Dacko-Pikiewicz, Dean of the Faculty of Applied Science, Professor Katarzyna Szczepańska-Woszczyna, and Supervisor, Professor Tomasz Gąsior, and the whole WSB University Senate, and acting pursuant to the resolution thereof of 26 September 2025, I confer the title of Doctor Honoris Causa and the associate rights and privileges upon Professor David Fisher, an outstanding scholar of global renown in the fields of medicine, molecular biology and oncology, professor at Harvard Medical School and head of the Department of Dermatology at Massachusetts General Hospital in Boston. In recognition of his scientific breakthroughs in the fields of cancer biology, immunology, and skin pigmentation, his extraordinary ability to combine fundamental research with clinical practice, his dedication to patients, and his role in inspiring generation of scientists and doctors, a man of great wisdom, scientific passion, sensitivity, and humanism, where the rigor of science and the poetry of music find the common with. Signed, Supervisor, Professor Tomasz Gąsior, Dean of the Faculty Applied Science, Professor Katarzyna Szczepańska-Woszczyna, and Director, Professor Zdzisława Dacko-Pikiewicz. Most distinguished professor, with regard to the conferment of the title of Doctor Honoris Causa of WSB University and the associated honors and privileges, I present to you the epitagium and the diploma bearing the seal of WSB University.
The university flag, come in. Ladies and gentlemen, please be seated. The letter of laudation on behalf of Professor Marek Gzik, the Secretary of State and the Ministry of Science and Higher Education, will be read by Professor Katarzyna Szczepańska-Woszczyna, Vice Rector for Science and Education at WSB University.
Dear Professor Fisher, I'm delighted and honored to offer you my heartfelt congratulations and being awarded honorary doctorate by WSB University in Dąbrowa Górnicza. Your professional career, spanning many years of research, teaching, and organizational work, set the example of the entire academic community. In awarding the title, WSB University pays tribute to you, a physician and a scientist, a leader, a teacher, and an outstanding musician. Your contribution to the development of modern medicine, molecular biology, and genetics deserves the highest recognition. Your research, which has led the groundbreaking discoveries in the treatment of melanoma, one of the most aggressive forms of skin cancer, is particularly admirable. With your commitment and passion, you have expanded the boundaries of our knowledge and created the new possibilities for prevention and treatment. I cannot be overlooked that your discoveries have been widely published in leading scientific and medical journals such as Science, Cell, Nature, and the New England Journal of Medicine. While examining your professional career, in the worth highlighting your extraordinary achievements in teaching and mentoring, you've educated many generations of students, inspiring them to pursue careers in medicine and scientific research. Doctors and scientists around the world draw your skills, experience, and expertise, and then use them in their work in academia, biotechnology, and clinical medicine. Your authority is your evidence by the honorable and responsible position you have held, to name just Chief as the Massachusetts General Hospital Department of Dermatology, Director of the U.S. Department of Defense's Melanoma Academy, member of the U.S. Nation Academy of Medicine, Fellow of the American Association for Cancer Research. Finally, I would like to mention another wonderful passion of yours, which is music. Your success in the field are also to be commended. As a professional cellist, you have performed on numerous prestigious stages, playing with orchestras and chamber ensembles, sharing your musical expression and creativity with audiences around the world. Once again, I offer my sincere congratulations on this honorable distinction and wish you satisfaction with your work, every success, and continued outstanding achievement. Respectfully yours, Marek Gzik, Secretary of State, Minister of Science and Higher Education, Republic of Poland.
Thank you, Professor. Ladies and gentlemen, it's a great day for the whole WSB University and for all Akademia. And I kindly invite to the distinguished guests and delegations who wish to extend their congratulations and expressions of respect to Professor David Fisher, distinguished Dr. honoris causa, to do so immediately after the ceremony in front of the Assembly Hall, please. Ladies and gentlemen, Professor David Fisher is not only an outstanding scholar, but also a gifted cellist. Let us now give the floor over to music, Professor Fisher's other great passion. On the stage, we shall welcome the musicians of the Karol Szymanowski Philharmonic in Krakow, the Philharmonic Quartet, accompanied by the renowned soprano, Miss Magdalena Drost. The program includes timeless masterpieces that have stirred hearts and imaginations for centuries. Among them, there are Spring from the Four Seasons by Antonio Vivaldi, the aria Vidit Sum from Stabat Mater by Giovanni Battista Pergolesi, Liber Tango by Astor Piazzolla, O mio Babino Caro by Giacomo Puccini, as well as a movie soundtrack by Ennio Morricone and Henry Mancini. May this sound serve as a symbolic tribute to Professor David Fisher, a scholar who, through his work and discoveries, opens new horizons in medicine and biology, uniting scientific precision with humanistic sensitivity, but also to the man in whom the rhythm of research and the rhythm of music form a perfect harmony, to the scientists whose passion and wisdom inspire generations of physicians and researchers. Ladies and gentlemen, may music today become our shared language of gratitude, admiration, and emotion.
dream maker, your heart breaker. Whenever you're going, I'm going your way. Two drifters off to see the world. There's such a lot of what to see. We're after the same rainbow sand, waiting around the bend. My huckleberry friend, moon, river, and me. Two drifters off to see the world. There's such a lot of what to see. We're after the same rainbow sand, waiting around the bend. My huckleberry friend, moon, river, and me.
Let's give a standard ovation for the Philharmonic Quartet and Miss Magdalena Drost. Please stand up, please stand up. Once again, a deep thank you to all of you, to the Philharmonic and to Miss Magdalena Drost for this wonderful performance. So after this performance, we can say that music has become our shared language of gratitude, admiration, and emotion. Thank you once again. And now, I have the great honor of inviting Professor David Fisher, Doctor honoris causa of WSB University, to deliver his lecture.
Thank you. Thank you so much. I'm moved beyond words by the experience this morning from the moment we walked into the room and the beautiful singing from the choir, which grabs you by the heart and set the stage for a remarkably emotional event. All of the words that have been spoken this morning are so deep and so thoughtful, and they grabbed, in many cases, the essence of what for me was simply a journey I will share with you in a few moments, but something that when I look at it from outside in and I hear the remarkable words, it actually seems I have to pinch myself and say, was this really me? It's quite amazing, and to receive this kind of enormous honor and respect from such esteemed colleagues here, I cannot thank you enough. So I express my gratitude and my sincere appreciation. So this is the title of my address, and I'm going to try and share with you over a short period of time the journey that I took in my professional career. And I will very briefly describe a little bit of my own family's history. My parents were both born in what is now Poland. My family is Jewish, and both of my parents were born in what was previously Breslau, now is Wrocław. Their parents had worked in Kępno and Opole. Both of my grandfathers had graduated from university in Breslau and in law. One of them actually became a judge and also was a philosopher. Very sadly, on Kristallnacht in 1938, both of these grandfathers were arrested. My mother's father was taken to Dachau concentration camp. My father's father was taken to Buchenwald concentration camp. Miraculously, they both managed to get away, or I would not be here. And fortunately, over many, many, many years, we were able to survive, and our family was able to rebuild. And then many, many years later, in 2008, when my father turned 80 years old, he brought the entire family back. And we visited Poland, Czech Republic, multiple cities, including Wrocław. And I have to say that while this was a shattering experience in some respects for our family, we saw locations where extremely painful things happened, unfortunately to many members of our family who never made it out alive. On the other hand, we saw a transformation, and I must say that the feelings that we have had more recently, and absolutely the feelings that we have had on this trip, my wife, who is accompanying me here today, and in my case, I cannot tell you the message that it brings to us way beyond science, even way beyond music. There is a message of hope and of freedom and of respectfulness that I think our world terribly needs. I wish we can broadcast this wide and far. We live in a very difficult world today, but this is the message of hope, which for me gives us optimism and friendship and collegiality. We have much more serious enemies like cancer, like disease, like sadness that exists throughout the world, even conflict. Those are the real enemies we need to overcome. And when I see this kind of reception and this kind of respectfulness in the way we interact as people, I feel optimism, enormous optimism, and I want to thank you for sharing this. And I feel this in the music. The music we heard this morning is extraordinary music. This was not just a quartet with a soprano. This was top of the line, fantastic music. I would hate to say I wish I were playing, but I loved listening, and I know you share the emotion. This cuts straight to the heart. This goes through all religions, all peoples, all cultures. It is beautiful, it is touching, and it is optimistic. So thank you so much for that. And just the same for the singers, who are equally magnificent. So I'm going to share with you a journey for me. As I mentioned, it is fascinating to step out of the box and look back at something which for me was, what's the next question? What's the next problem? Where's the next opportunity? And I want to emphasize that when you do research, biomedical research, and when you ask what you believe are some of the most important questions, they're really difficult questions. And one problem with my presentation, as I put my slides together for this presentation, is that I will actually share a little bit of scientific data with you. I will try very hard not to make it too complicated, but it kind of looks like all the experiments worked. They did not work. The majority of them don't work. And in fact, when you ask difficult questions, they shouldn't work. If they all work, you're asking questions that are too simple. You have to be willing to have the resilience to get up when it doesn't work and scratch your head and try to understand what was the problem. Was it a technical problem? Was it a conceptual problem? And try to learn and grow. And I would say, as I have looked back in my career, and I've shared this with many people who have worked with me, many trainees and students, is that I would think most of the biggest discoveries that have come from our lab that we've been fortunate to have were accidents or mistakes or confusing results that we thought did not work, but then when we stepped back and thought about it further, actually there was something profound and interesting that we could learn. So it takes a certain humility, a certain stubbornness, a certain confidence to move forward. So as I quickly go through this journey, I do want to emphasize this wasn't easy, and for every experiment that worked, there probably were 5 that did not work, and this was part of the journey as well. So it actually began with a mouse that had a white spot on the belly. The normal mouse should be completely black, but this mouse had a mutation in one gene, only one out of 25,000 genes in the genome, one mutation, and it caused the belly to be white. That's when it had one normal copy of the gene, plus, and one mutant copy, minus. But if you crossed 2 siblings so that you had a mouse where both copies of the gene were mutant, the mouse came out completely white. And you may notice its eyes were very, very small. So this mouse was called microphthalmia, small eyes. And the gene, which we later determined to be something called a transcription factor, regulates the expression of other genes. The gene became known as the microphthalmia transcription factor, or MITF. This is where my scientific journey began, because this mouse spontaneously developed, but nobody knew what the gene was that caused this white color, the white spotting. And I was fortunately in the right place at the right time. Somebody approached me and said, could you study this? And we happened to have the right experimental test to study what this was. So if we skip forward 25 years, we now know that the MITF gene encodes a protein which regulates the production of pigment. It is the fundamental, people call them now the master regulator of the entire ability to make melanin, all the different kinds of melanins, different colors, different types in your skin, in your hair. This is the master regulator that controls a whole pathway, a complicated pathway to produce pigment. And it also is regulated by other factors such as ultraviolet radiation. When you tan, it goes through MITF. So this sat in the middle of intricate, complicated processes that are very prominent. You see in a person their pigmentation immediately. It's one of the most prominent things we see. We see their skin, and we know that this relates to cancers, it relates to beauty, it relates to self-confidence, how we look, how we feel. It's right in the middle of things. And this was an interesting 25-year journey. And I'm going to take you a few steps of the way and even show you some of the experiments that led us to some of these conclusions. So this is the gene. It's called MITF. So firstly, I'll describe for you pigmentation. How did we discover that this plays such a central role in pigment production, as well as the transfer of pigment out of melanocytes? Because it turns out that the pigment, although it is made by a melanocyte, the cell that is a factory of melanin synthesis, it doesn't stay there. When you look in your skin, that is not melanin in melanocytes, it's melanin in the other cells of the skin called keratinocytes. They're much more abundant. So the melanin is produced in melanocytes, and then it is transferred out in this process that is demonstrated here, as though these little pockets of melanin are shipped out to keratinocytes, and I will show a bit about this. So very, very briefly, the first thing that we could identify is that the protein encoded by the MITF gene could bind to DNA, and it had a specific sequence of DNA that it could recognize. And the sequence it recognized had this sequence CACGTG. How did we prove this? Because we could take radioactive DNA. This is many, many years ago. We don't even use radioactive DNA anymore. But at the time, we used radioactive DNA. We had a source of the MITF protein. And when we mixed them together, the free radioactive DNA was shifted to a different mobility in the kind of a gel matrix where we looked at this. This was called a gel shift experiment. And if we added non-radioactive DNA with this sequence, it competed away the radioactive interaction, radioactive DNA with the protein. And it did it selectively, because if we had DNA with a change in these two positions, instead of CAC-GTG, it was GAG-GTG, which it does not bind. MITF cannot bind. That could not compete away the radioactive DNA. Only CAC-GTG could. Or it turned out CAT-GTG could as well. This is a very small change between the C and the T. This turned out to be a very simple looking experiment, but it turned out that every gene involved in the production of melanin has either CAT-GTG or CA-GTG sitting right at the beginning of the gene. And not only was this present in all these different genes, it was present across species, in human DNA, in mouse DNA, in fish, in birds, across many species. So evolution conserved this little 6-nucleotide sequence, and it is exactly the sequence that MITF protein could recognize. And in fact, if the MITF protein is added, the gene could be stimulated in its expression. Not only could MITF bind there, but it could turn on the gene and stimulate the production of RNA from that gene, which turned out to be all the pigment enzyme genes, all the factors that transport the melanin from the melanocyte to the keratinocyte. So this was sort of the birth of the idea of how melanin is produced, and that there was this one dominant factor, MITF, that is in the middle of stimulating and regulating the entire pathway. So we now could see, and there's enormous work from our lab, but also many, many other labs around the world that help to establish this pathway. MITF regulates pigment production, and the transfer in these little vesicles, we call them melanosomes, eventually out of melanocytes into keratinocytes. I'll show you pictures of that in just a moment. How is MITF itself regulated? Because we know pigment is sometimes turned on, such as when you go out in the sun and you can get a tan, if you can get a tan. How is that involving MITF? What is going on? So the assumption had been, including my own assumption, that ultraviolet radiation must penetrate and somehow turn on MITF. Because we know if MITF is not there, no pigment can be made. You saw white spots, completely white mice. You have to have MITF work. So presumably, if ultraviolet radiation shines on your skin, it penetrates through and stimulates MITF somehow. However, there was a clue from humans and genetics that suggested that model somehow does not make sense. And the reason is that red-haired people and red-haired mice, I will show you in just a moment, don't tan. We all know this. In fact, not only red-haired people, even light-skinned people, myself, I don't tan so well, but red hair is the extreme example of this. And this didn't fit because we know that ultraviolet radiation can penetrate into a cell and it doesn't care. And I should say, this is the gene, MC1R, right here, is the name of the gene that is affected in red-haired individuals. One gene, in vast majority, over 95%. And so we know, on the other hand, that ultraviolet radiation doesn't care what proteins are on the surface of a cell. It penetrates right through. So it did not make sense that depending on the state of this protein, red-haired version, black-haired version, that you would tan or not tan, ultraviolet radiation goes straight through. We wondered, does this tell us that actually ultraviolet radiation is stimulating a different cell to make a protein that activates this receptor? And then that somehow activates MITF. So that if you have a version in a red-haired individual that cannot become activated by a different protein, then this pathway is blocked, like a brick wall in that position. That was the hypothesis, just looking at human data because red-haired individuals cannot tan. And this turned out to be the case. I will show you, here are black-haired mice where the MC1R protein has very strong response. And as you look at the ears with UV radiation, you can see the ears get darker and darker as they receive UV radiation. But in red-haired mice, which are identical, 25,000 genes identical, except for one, which is the MC1R gene that does not work, just like a model of humans. And you can see their fur is red-haired, very similar to humans. And you can see even at the highest doses, their ears don't tan. So this was a very complicated experiment to do in mice, but we knew it would be true because human red-haired individuals don't tan. So we knew that this would very likely work, and we could see that in the mice this was the case as well. So we then asked a more complicated experiment, and I promise I'll go through this quickly. Could it be that if you take keratinocytes, which are the much more abundant cells in the skin, in the epidermis, and separately in the laboratory, UV radiate those, there are no melanocytes there, and then you take the media, the liquid that these cells are growing in, and you separate it from the keratinocytes, and you add it to melanocytes, was there something made by the keratinocytes that could stimulate the melanocytes to turn on MITF? And there was. When the keratinocytes were not treated, MITF stayed low, but if they had received UV radiation, MITF went on very strongly. But if you removed chemically the protein that stimulates that MC1R receptor, we could see that that activity was gone. And lastly, we could see this worked in melanocytes or even in melanoma cells. It could stimulate MITF production. And it worked in mouse keratinocytes, it worked in human keratinocytes. So this smelled like actually UV does not target melanocytes to make pigment, it targets keratinocytes to make the protein that will stimulate melanocytes to produce pigment. And this pathway requires the receptor that red-haired individuals don't respond with. That protein in redheads doesn't work. And I should emphasize, I don't see any red-haired people in the group here today. Usually when I speak, there's one or two. we never refer to red-haired individuals as mutant, because in fact it's not mutant, it is a variant, it is a normal variant. In fact, it probably was a beneficial variation in human populations, especially as humans moved away from the equator, where the sun is weak in the winter, and it probably benefited for the production of vitamin D because ultraviolet radiation can contribute to vitamin D production. I will talk more about that in a few moments. So here was a pathway that placed the tanning response into a biochemical context. So in fact, ultraviolet radiation does not act directly on melanocytes, it acts on keratinocytes to produce the protein from which this MSH, melanocyte-stimulating hormone, is released, activates this receptor to produce intermediates and activate the production of MITF. And that is the pathway that produces the tanning response. And in red-haired individuals, this protein does not respond to MSH, the tanning pathway is interrupted. Now, a very rigorous proof that this response depends on the signaling from this MC1R protein was to ask whether we could take mice that were red-haired and had always been red-haired, and their parents and grandparents, their whole ancestry was genetically red-haired, and ask a very simple question. Could we find a drug that would activate the pathway right after MC1R? MC1R was not working. Therefore, they were always red. MITF levels were very low, and the production of pigment was only red-blonde pigment, never brown-black pigment. We wondered if we could take a drug, and we found a drug called forskolin that stimulates cyclic AMP levels to come on strongly. And it was a crazy experiment, but we took red-haired mice, and we topically applied forskolin on their skin, because we thought, if our model is correct, maybe the red-haired mice would upregulate MITF and actually become darkly pigmented. And this is exactly what we saw. These are all red-haired mice. All of them are the same, but every other mouse received the topical forskolin drug, and as you can see, they became dramatically darkened, and you could see pigment in the skin. And I want to emphasize one thing, which is that when the pigment is made after forskolin, and I should say when it's made normally in human skin or mouse skin that are genetically black, the pigment is not just dispersed randomly in the keratinocytes, but it actually sits like these little umbrellas over the nucleus of, you can't even see, but right under these little arcs of melanin, there is the nucleus of the keratinocytes. That's where the DNA sits. And the DNA is extremely sensitive to mutations from ultraviolet radiation. So if you wanted to protect your skin cells, one of the most important places to protect is the DNA in the nucleus. And evolution has actually produced a trafficking, a localization of the melanin in these little umbrellas over the nuclei. And we saw that even with just forskolin, this very artificial way of stimulating pigment, that the entire pathway was true to what you would see in a genetically black mouse or genetically dark pigmented human. And so this is the pathway, and we could see that indeed forskolin was able to produce real pigment. And in fact, if we challenged the red-haired mice with ultraviolet radiation and we measured skin cancer production, we could see a dramatic protection, a delay. This is a mouse that actually has a mutation that makes it very, very sensitive to ultraviolet radiation. Most people are not this sensitive, except a few people have these mutations. It's a rare condition. But the darkening was able to produce a long, long delay, very significant benefit. And this led us to the idea that was mentioned earlier, perhaps dark pigment might eventually, if we can do this in human skin, serve in a protective activity, a new way of trying to protect against skin cancer from ultraviolet radiation. As I mentioned, the pigment is produced because of MITF, and then these melanosomes are trafficked out to keratinocytes, and as shown in this cartoon, it doesn't randomly distribute in the keratinocytes, it's over the nucleus, and it coalesces in a way that is like an umbrella that you could see in that previous picture. But another point that was also made earlier in the introductory comments is that sitting right next to the production of melanocyte-stimulating hormone is another peptide called beta endorphin. And endorphin has become quite famous because endorphin is our body's natural opiate. And we've never completely understood what endorphin does in humans. Some people describe how when you run, you feel a high, you feel good. In many situations, perhaps endorphin gives you euphoria or a feeling of satisfaction. But here we saw that endorphin was going to be made every time ultraviolet hit the skin. If you are able to get a tan, you must be producing endorphin. It's part of the same intermediate. It has to be made at the same time. And this led us to wonder whether the endorphin might do something. Is it just being made? Is it just sort of a biochemical detail? Or is there an impact behaviorally? And we were not psychologists, we were not psychiatrists, but one of the beauties of being in an institution like Massachusetts General Hospital is that just upstairs from us, there were people who were experts in pain and opiates and this sort of thing. And therefore, we could study whether this affected opiate pathways such as pain sensitivity. As you know, opiates like morphine are very powerful at suppressing pain. And so we could look at pain, and we could look at other behaviors because unfortunately in the modern time, the worst problem with opiates is addictive behavior. And many people in society, unfortunately, are plagued by problems of opiate addiction. And so we wondered whether endorphin might be doing something like this. And I will show you a couple of experiments that address this. So even in mice, if we gave very low doses of ultraviolet radiation, we didn't want to give nuclear doses of ultraviolet radiation, but doses below sunburning doses, very, very, very low. And we kept giving these doses over days. And we took, because remember, we're only radiating the skin, but to affect behavior, it somehow has to get to the brain. So first, we assumed endorphin would need to be made sufficiently that it would leak into the bloodstream and that we should be able to measure something in the blood. And sure enough, after about a week, blood levels of endorphin started to rise. And when we stopped giving ultraviolet radiation, the blood levels of endorphin fell right down quite quickly. But also, if we measured other behaviors, such as pain sensation, I'm not going to show you, they became numb. If we quantified in mice, if they could feel temperature, heat, pain, or pressure with pinpricks, it took a stronger signal. As we gave ultraviolet radiation, they couldn't feel it unless they got a stronger signal, and it correlated exactly with whether we were giving ultraviolet radiation or if we stopped. And then we did an experiment in which we gave UV, we saw that the endorphin level rose, and we kept it going for a number of weeks. 2 or 3 weeks, I don't remember exactly. And then abruptly, we blocked the opiate receptor. There is a drug called naloxone. This is the drug you would give somebody with an overdose of heroin, fentanyl. If you have a patient who has overdosed, you're afraid they're dying, you block the opiate effect instantly. However, we know in humans, if you block opiate signaling in an addict, they will have withdrawal symptoms. They will shake. They will have a whole bunch of horrible symptoms, which is part of the reason they continuously take the opiate. They are dependent on the opiate. And in the mice that were only receiving ultraviolet radiation, the mice where they suddenly received ultraviolet radiation plus naloxone started to shake and tremors and jumping and all the symptoms that in a mouse are the equivalent of a human withdrawal symptom. And we could see this in the mice. And I would also, I don't have the data here, if we removed the gene for endorphin, we could genetically remove it, you could give as much UV as you want, they wouldn't have any of these effects. They would not have withdrawal symptoms, their pain threshold wouldn't change. So we could track this to endorphin being synthesized in the skin because of ultraviolet radiation. So this suggests that there is an addictive response to UV, one of the most powerful drives to seek sun exposure. Now, from the perspective of a dermatologist, this makes no sense. should we have evolved to want exposure to ultraviolet radiation, which causes more cancer than probably anything on the entire planet? It's the most common carcinogen there is. It seems absolutely backwards. Why would this be? And when you look at science and you see something that is so completely backwards, there has to be a reason. This could not be an accident. So what would be the reason for this? We wondered. So we had a hypothesis. There is only one thing in evolutionary times, not today, and I'll emphasize this in just a moment, where ultraviolet radiation actually was good. In fact, it was required for our health. That was the production of vitamin D. 50,000 years ago, 100,000 years ago, it was very difficult to find vitamin D because our body lacks, and I should say not only us, the entire animal kingdom has no enzyme to carry out one of the many steps in the production of vitamin D. But ultraviolet radiation has that activity in a very specific portion of the ultraviolet spectrum. So when ultraviolet radiation hits our skin, a certain carbon-carbon bond is cut, and now vitamin D can be produced. But in the complete absence of vitamin D, you will become vitamin D deficient, and it is lethal, it is fatal in childhood, a disease called rickets. So we cannot live without this. So we wondered, could this behavior help to protect individuals from vitamin D deficiency by having vitamin D be part of endorphin production? You get a reward for going outside in the sun. Imagine you live in Scandinavia 100,000 years ago, and in the wintertime, there's only 15 minutes of sunlight. But you live in a cave because it's freezing cold, it's dark in the cave, and you can either stay in the cave all winter long, but you will die of rickets, and your children will die of rickets, because without vitamin D, we don't absorb calcium. Without calcium, we cannot make bones. You cannot live. So if you had a behavioral inclination to go out for that little bit of sunlight, and you feel euphoria, you feel really good, that's the behavioral drive, it may save the species. It may rescue through a behavioral effect. And of course, the converse of that is if you stay in the cave, you may feel depressed in the winter, you may feel miserable, which unfortunately happens as well. We haven't verified that this is through vitamin D, but it wouldn't be surprising that this pathway works in that fashion. So we did a behavior experiment. This is a psychology experiment with mice, because it's actually very difficult to establish an evolution argument in the time frame of one graduate student or a postdoctoral fellow, because it took 100,000 years to do this, and my students really like being in the lab and I love having them, but 100,000 years is way too long. So we had a hypothesis that we wanted to test to try to establish a connection between maintenance of vitamin D levels and the UV response and endorphin. And so could vitamin D somehow regulate the response to opiates, to endorphin? And we had a hypothesis, which is maybe if your vitamin D level is very low, maybe you would feel very powerful euphoria. So if you're in the cave in the winter, your vitamin D level has gotten low, low, low. You go out for 5 minutes, you feel great. Whereas once you get the exposure to UV, your vitamin D level becomes normal. Now it doesn't feel so good anymore. Vitamin D may regulate the opiate response behaviorally. And if that would be the case, we felt this would establish a connection that would make evolutionary sense. So how did we test this? We test this by having a setup with 2 boxes and a little tunnel in between. And we could give mice UV radiation and naloxone to block the behavioral response. But essentially what we're asking is, do they feel the UV, and do they prefer to be in the cave where they receive UV? Do they, you probably heard the experiment where mice would push a bar and receive morphine, and they love it, and they would push it till they die because they're so addicted, it feels so euphoric. Well, we were asking a similar question, but with ultraviolet radiation. And we asked, is it true if they're vitamin D deficient or if their vitamin D is normal. And what we found is if vitamin D was normal, they didn't really care whether there was ultraviolet radiation. But if the vitamin D was deficient, now they, If the vitamin D was deficient, the mice had a huge preference for ultraviolet radiation. So the effect was enormous, and it turned out, I don't show it here, again, if endorphin gene was missing, they didn't care. They didn't see this behavioral benefit of the euphoria. And so this pathway appears to be accurate, that this is not only regulating pain, but it is regulating behavior. And we think that this underlies UV-seeking behavior, which is one of the largest problems we have with skin cancer now, because people who are highly educated, who understand the risk of sun exposure and the association with skin cancer, on a beautiful, sunny Sunday in the summertime, still want to go to the beach because it feels good, or go out to play, not necessarily remembering to protect their skin. And this is organic, it's in our genome. So this is part of the challenge that we have, and I think also a prevention opportunity. So you might ask, what would happen if vitamin D deficiency is amplifying the euphoria? Could you actually push this the other way? Can you reverse this effect? We did a study where we looked at opiate addicts in Boston. Many of them were vitamin D deficient. It fit the model. And in mice, we asked if mice are vitamin D deficient and have this very high euphoria from opiates. Now forget UV for a moment, but morphine, heroin, these kinds of. What happens if you correct the vitamin D deficiency? Could you reverse this opiate-seeking behavior, the euphoria that they get? And in fact, we could see in the mice this was reversible. So we have not carried this forward yet, but in fact, this is potentially a treatment for opiate drug addiction. For patients, if you gave extra vitamin D, their perception of euphoria from opiates, part of the drive for feeling good and dependency, perhaps at least a portion of that might be reversible. Vitamin D is inexpensive, extremely safe. What a wonderful opportunity to learn from skin about a potential treatment for prevention or for management of opiate addiction. So I've described much of this pathway, including MITF, and as I close my talk, I want to mention that despite all these pathways that I've been describing to you, we could see pigment regulation, response to UV behavior, but we knew something much more was going on with MITF than the regulation of pigment. And the reason for that is that when we look at these MITF mutant mice, and by now we knew that this also happens in people, and it's not so rare, actually. There are conditions, if you have ever seen somebody with a white, we call white forelock, this is the equivalent of a white belly spot in mice, when you see it along the midline, the ventral surface, that this area, it's not melanocytes that don't make melanin, it's actually no melanocytes are there. So the melanocytes have actually died. MITF is not only regulating pigment production, it is regulating the survival of melanocytes as a cell. So something vital about the viability of melanocytes requires MITF function. And that's much more profound and led us to ask some other questions that I'll very briefly describe to you. One of them was, we found an MITF mutation in mice where the mouse progressively became gray and then completely white. And this was caused by a mutation, a mild mutation in the MITF gene. If it were strong, the mouse would have been born white, but it was weak, so the mouse was born with quite a bit of pigmentation, and as it got older, it gradually turned gray and then completely white. So when we looked in the skin, the melanocytes had been dying. They were going away. So again, the melanocytes need MITF, and they even need it after birth, not just during development. So it is required to maintain the survival of melanocytes. And we could see a variety of mouse strains that have this gray phenotype, gray hair phenotype. And here was an example of a gene called BCL2. I won't go into detail, But we knew that MITF regulates the BCL2 gene. So perhaps when MITF is not functioning, BCL2 is not functioning, and this could be part of why melanocytes cannot survive. And perhaps it could even relate to melanoma. Now we're getting to life and death, which is a problem in cancer. So we're moving beyond just controlling pigment, but to the survival and of the melanocyte lineage in different ways. When we looked at BCL2 mice, we actually discovered something quite profound, which is that in postnatal day 8.5, this is day 8 after the mouse was born, between day 6.5, this is a mouse that has no BCL2 gene. This is a mouse that became completely white within about a month or two after it was born. We can see that at day 6.5, you see these little blue dots. This is a group of melanocytes called stem cells. I won't go into the specifics of them. But just 2 days later, at day 8.5, this entire population of melanocytes has disappeared. They're gone. When BCL2 is not there genetically, this population of melanocyte stem cells is gone, and you need these cells at the next hair follicle cycle to produce melanin so that the hair would be darkly pigmented. And that is the phase where all the new hairs come in and they're white. So this was the first example where mechanistically we could understand where gray hair comes from. Now, people don't really care so much about why mice have their hair turning gray. They're much more interested in why humans have their hair turn gray. And so we have studied this. I'm not going to go through the details of it, but it turns out as we age, it's exactly highly similar mechanism. We are losing our melanocyte stem cells. And as we age, we could recognize, this is 20 years ago we published this paper, that the melanocyte stem cell number is gradually becoming lower and lower in each hair follicle. And when it reaches the critical point where there are none left, the next cycle of hair growth, the hair will come in completely white. So gray is a mix of black hairs and white hairs, and eventually there are no black hairs left because all the melanocyte stem cells are gone and the hair comes out completely white. Why do we lose melanocyte stem cells? And amazingly, because we had heard this from our parents and friends for many years, it is actually stress. And we studied this in a paper that was just published a few years ago, where you could inflict different types of stress in mice. Some of them were painful stress with a drug that actually caused a significant amount of pain for a few moments. In other cases, it was mild stress like social isolation. They'd have to live alone. Mice like to be together. In either of these settings and other types of stress, the kinetics of hair graying was amplified, and we could see exactly the same mechanism. It was the loss of melanocyte stem cells that was actually responsible for the hair graying phenomenon. So this is in fact a clue of why our hair turns gray. And the mechanism behind this is fascinating because perhaps there even are strategies to apply to melanoma where you want to kill cells of the melanocyte lineage, melanoma cells. And so these are areas of great interest to us at this point. Now, I'll finish with, here we were looking at loss of MITF, loss of melanocytes. Are there any situations where MITF is overactive? And is it possible when it's overactive that it affects survival of melanocytes in the other direction? There are too many of melanocytes. Could this be participating in the formation of melanoma? And we found this in two different ways. We identified biochemically that MITF is modified. Its protein structure is changed by the action of a number of different proteins in the cell, which have separately been identified. Each one of these red arrows identifies something that we call an oncogene established to be causative in human melanoma. And we could see that they converge on regulating MITF. So not only does MITF regulate pigmentation, it regulates the growth, proliferation, and the survival of melanocytes. And we can see that this also occurs in melanoma, because in fact the MITF gene itself is sometimes dysregulated. In this case, it is dysregulated because the genome of the DNA in the nucleus of a melanoma cell sometimes has extra copies of certain genes. And here you can see This is a fluorescence examination. These 3 cells are melanoma cells. And in red, you can see that for this gene, this is a control gene, we should only have 2 copies of every gene, one from our mother, one from our father. All of our cells should inherit 2. And when we look at how many copies of the MITF gene, you can see there are way more than 2 copies. This is an example of the MITF gene being genomically amplified. That is sufficient, too much activity of MITF to confer too much survival, too much proliferation. And MITF now is well known to be a human oncogene, either by too many copies, or what we found a few years later is that there are mutations, a very specific mutation in MITF, and this is a large family tree where each of the red letters, E318K, these were individuals where melanoma was all over the place, often at young ages, often multiple melanomas in individuals. And this could be traced to a very specific mutation in MITF that overactivated the protein. So we could see either too many copies of MITF or mutations that overactivate MITF were causing melanoma in humans, and this has been examined in other strategies. And from this information, we could identify other genes that are highly related to MITF, TFE3, B, and C, and we could see that now it is well established that these also, when they're overactive, they cause not only melanoma, but a kind of cancer called sarcoma, they cause kidney tumors, different types of sarcomas. So this is a whole family of human oncogenes that came. And from this is related melanoma therapy. We know that there are different types of therapy, targeted therapy, and immunotherapy, which is extremely important. When the immune system can be activated to target melanoma cells, and this is the big revolution in the therapy of melanoma, and I would say the most exciting revolution in the therapy of cancer, perhaps for the last century. It's very, very important, and it is scientifically grounded. It is not by any means only my work, it's the work of countless people in academia, in industry, clinical trials, patients who have contributed to this knowledge. It turns out that why would melanoma be the cancer that is so particularly responsive? And it appears that when ultraviolet radiation mutates the genome of a melanoma cell, many proteins have a change in their sequence from that mutation. Even if that change doesn't cause the cell, some of those mutations cause the cell to grow more, but many others are silent. They don't cause the cell to behave differently. But because the mutation is causing a protein to look different, the immune system looks at these slightly different proteins and says, this looks foreign. This is not my normal protein. And so the immune system recognizes these are called neoantigens, mutated proteins that are private for each person's melanoma. And one of the important studies that we contributed is that we can see that when the immune system tries to attack the neoantigens, the recognition of attack, especially with the best immunotherapies, spreads in something we call epitope spreading to attack even normal melanocyte proteins, so that the ability to activate an immune response against melanoma is essentially an autoimmune disease, but it is a beneficial one because the melanoma cells will die before all of our melanocytes die, and then you have a benefit, and patients can be cured of melanoma. We are now at a time for patients with metastatic melanoma where 30 to 50% of these patients can be cured, stably, durably cured with immunotherapy. And these therapies are being addressed for all cancers. Most cancers don't respond that well, but there are ideas of how to get the autoimmune activation in cancer cells that don't have so many mutations to rescue the immune response and perhaps benefit patients with other types of cancers. We think in the prevention category, if there were one cancer that should be highly preventable, we think melanoma should be there, because how many cancers do we know the cure as clearly as we do, the cause, how many do we know the cause as clearly as we do in the case of melanoma? Yes, for cigarettes, we know for lung, but most cancers in humans we don't. Ultraviolet radiation, we really do, and our progress has been weak in preventing melanoma. Endorphin may be part of it, that you feel good, and so there's that challenge. we think that there are other challenges. We think sunscreens, although we highly recommend them, they're missing some components of different types of melanins, as we heard earlier from Thomas, are themselves, some melanins, the red pheomelanin, has a chemical activity that is mutating the genome of cells. There's more information, and we think optimistically there will be more opportunities to do better with prevention. So a journey that began with a spotted mouse brought us to pigment biology, tanning response, addiction, hair graying, human oncogenes, immune attack approaches, and hopefully new ways to be attacking the problem of prevention. So in my own case, I am the product of a journey in which my family left to save their lives, and I return today with enormous pride, with gratitude, with humility, and my thanks to my enormously gracious hosts, saluting their commitment, your commitment to science, medicine, and to humanity. This has been an amazing journey for me. I feel immensely honored, and I look forward to continuing collaborations with you all. Thank you very much. I do have 2 quick slides just to emphasize. So this is a photo of my current lab. This is actually from last year. I must emphasize Dr. Konrad Kaminiu. I think I hopefully got it. Okay, good. Who is here and is a brilliant young physician training here and host for me. We were very fortunate to host Konrad in our lab over the last year. He contributed to very important studies on red-haired models and nevi and again, this issue of diagnosis and prevention of melanoma. We're grateful to have him, and he has been a magnificent host. And also, just to mention, 2 months ago, we had a reunion of all people who have been in my lab over many years. And this was a very large group who came from all over the world, and it's enormously important for me to acknowledge they are the people who did the work that you just saw. They're the people who have stimulated me and helped me to answer some of these very difficult questions. That is one of the greatest joys in the honor of carrying out biomedical research, is learning from the people who you are working with on a daily basis. And with that, I would like to thank you, and again, thank our gracious hosts.
Thank you, Professor Fisher. Thank you. Ladies and gentlemen, now Professor Łukasz Słukowski, President of WSB University, will address the assembly. Please, Professor.
Thank you. Thank you very much, Professor Fischer, for your wonderful lecture. I learned a lot. I know I have more gray hair. It's just a job of the academic manager. I understand why I love sun, so thank you very much. But actually, I have one reflection that is connected to my field, social sciences. That is roughly storytelling. Unfortunately, it's not like STEM, not like the medicine, but this reflection is about one of the great minds in the field of the behavioral economics, one of the creators of this field. I mean, Amos Tversky from Tel Aviv University. Together with David Kahneman, they created this discipline, yes, that they built it. And David Kahneman, 20 years later, get the Nobel Prize of the Swedish Bank from economics. And unfortunately, Amos Tversky couldn't do that because it's not post-mortem prize. But he died quite young, melanoma. And this is just a great, great hope. This is the development that is changing the humanity. That gives us a lot of hope, and we are very, very grateful for that, Professor Fischer. Thank you very much again.
Ladies and gentlemen, please rise.
Professor David Fisher, rector, dear Senate member, all professors, our medical students, I would like to thank you very much for the participation in this wonderful ceremony, and I'm now officially closing the offering of the honorary doctorate for Professor David Fisher. Thank you.
Ladies and gentlemen, please remain standing. I hereby declare the ceremony of Professor David Fisher, Doctor honoris causa of WSB University, formally closed. The university flag will be now withdrawn.
Gaudeamus igitur juvenes dum sumus. Gaudeamus igitur juvenes dum sumus. Post iucundam juventutem, post molestam senectutem.
The Honorable Senate is leaving.
Nos habebit humus. Vivat Akademia, vivat professores. Vivat Akademia, vivat professores. Vivat membrum quod libet, vivat membra quae semper sint in flore, semper sint in flore.
Ladies and gentlemen, thank you for participating in this event.
Thank you for participating. So.